to Science at Cal on the shortest day of the year. This is the winter solstice. I think it starts at 11:11 in about 10 minutes. 9/11. I should know better because I'm an astronomer. I'm Dan Wertheimer. I'm an astronomer here at Cal. Before I introduce today's speaker, I want to tell you about next month's speaker. You know, Science at Cal is a monthly series, always on a Saturday. You can find out when the lectures are. If you go to the Science at Cal website, just Google Science at Cal. The next one is on January 18th. It's Christian Reichardt. He's going to be talking to us about the Big Bang and the cosmic microwave background radiation that came right after the Big Bang, that tells us a little bit about the early history of the universe and how he measured it at telescopes on the South Pole. And I think he's going to be talking to us not just about the Big Bang and the early universe and how the first stars and galaxies formed, but also a little bit about life at the South Pole, because I think he spent a lot of time at the South Pole. And that'll be interesting. The lectures next year, most of them will not be here. They're going to be at Mulford Hall, which is right in the building adjacent to this one, 159 Mulford. Not all of them will be at 159 Mulford, though, so it's best to check the Science at Cal website so you can figure out where to go. Also on that website, you can make charitable donations. And you can also join the mailing list, so you can get these periodic mail links to find out who's going to be talking when. Today, we are fortunate to have John Taylor, who's a professor here at Cal, and he's going to be telling us about fungi. He was chair of the Division of Microbiology here. He's co-director of the Computational Genomics Resource Lab. He's received a huge number of awards for teaching and all kinds of things. Please welcome John Taylor.
Thank you very much for both the introduction and the opportunity to speak in Science at Cal. And I want to thank the high school students from American High in Fremont. And so for some of my friends and colleagues and neighbors. And it was mentioned in the introduction that I was chair of the Division of Microbiology and co-chair of the Computational Genomics Resource Lab. And my co-director, Brian Skaskowitz, is here and happens to be my next door neighbor. So today I'm going to talk about fungi. And the title is Diversity, Society, and Models for Evolution. And I timed this, and there should be plenty of time. So if you want to ask questions during the talk, please do so. Sometimes you forget them if you don't ask it right away. Say, for example, if you have to write a report. Feel free to do so. So I'm going to start out with three questions. What are fungi? Where are they in the tree of life? And how do they affect society? And most people, I think, when are asked what's a fungus, they think of a mushroom. So here's a mushroom, Lepiodoprasa. There's actually a little armillaria sitting down next to it. And I want to point out, one thing I really want you to take out from this is that there's more to the fungus than just the mushroom. So here's a cross-section drawing. There's the mushroom. There's the mushroom. The second part are these spores. And the whole point of a mushroom is to get the spore release above the dead air layer next to the earth, so the spores will go. And the third part is this web of little threads. Here is in the soil, and this is the part that feeds the fungus. So by analogy, this is the tree, and the mushroom is the fruit. You want to make an analogy to plants, and the spores are the seed. So I want to say a little bit about spores first. This is another kind of mushroom. It's a hardwood, a wood rot fungus. So its web is in a tree, decaying tree. And I picked them because they make a lot of spores. In a year, they'll make 1 trillion spores. And in this photo, you can see the spores there. And if you think about it, it's a sobering thought. If 2 of those spores reliably made a new fungus, in fewer than 100 years, it would equal the mass of the Earth. So 2 of them can't reliably reproduce. If there were 0, go extinct. So on average, 1 out of 1 trillion spores makes Now, not all fungi are mushrooms, and they don't just passively release their spores and are blown away by the wind. Some actively shoot them in the air to get them up off the dead air to work. Here you can see a very high-speed camera photographing the spores, and then a computer telling you how fast they move, and the warmer the color, the faster. And instead of shooting the spores one by one, they shoot them to create an air current, which carries them a little further up in the air, so they're blown further away from the parent. So fungi, the diversity in fungi is all about getting your spores into the air. Sometimes into water, sometimes stuff, but mostly in the water. So we talked about the spores briefly, and the mushroom. Now I want to talk about the business end of the fungus. And this web is made up of little threads called hyphae. Together they form a mycelium. So here is a hyphae. Two photos of the same hyphae. One with light, reflected light, and one where the nuclei have been stained and have been excited with ultraviolet. So the first thing is they're tiny. They're 10 millionths of a meter in diameter, 10 micrometers in diameter. But the cells are like our cells. They've got all the organelles that we have. They're eukaryotic. And they've got actually two key differences, which I'll talk about again later. The one I'm going to mention now is they have a cell wall. Like plants, they have a cell wall. We don't have cell walls. They grow at the tip, and this is another one. So this is the tip of the hyphae. It's been stained to show you the cell wall, and at the tip there, a thing called the apical body, which fits in clover, that is involved in producing the cell wall, and it's going to grow across the screen here, and you can see it growing. So as it grows, it exports enzymes that break down biopolymers. So it could be starch, cellulose, protein, nucleic acid, fats. And then it imports the monomers. That's what it feeds on. That seems like a very different way than, for example, we feed ourselves. But it's actually not that different. So it exports enzymes into an external space and breaks down biopolymers. So we think that this is being done in an internal space, but we're really a donut. And our alimentary canal is really an external space of our body. We secrete enzymes into that, break down biopolymers, and then feed on the monomers. And the key difference is that we've got muscles and bones and a nervous system and sensory system to go around and find food to put in our reactor. Whereas fungi make trillion spores and hope that one finds a place to feed. And then when we're out of food, we have to go find more food. What the fungus does, it just grows a little further into fresh food and repeats the process. When it runs out of food, it makes spores. So each of these hyphae arrange themselves, organize themselves into a complicated thing, mycelium. You can see that they're cross branches in the older parts of the mycelium. This is a growth on a petri dish, which is a very even environment. So it looks really boring. It just goes out in all directions. If you think about it in life, out in nature, the shape of that mycelium would follow whatever the food is. If it's breaking down wood, it would follow parts of the wood, etc. Now, mycelium, the hyphae and fungus, they're old. Now, they're not as old as the start of the universe that you're back in January. They're not 13 billion years old, but they're about 1 billion years old. And the fungi are about 1 billion years old. And hyphae are at least 400 million years old. And this is a rendition of one of the earliest land plants. This is a plant before roots evolved. There are no roots on this plant. There's an aerial stem and an underground stem, horizon. And if you look in fossils, 400 million year old fossils, my colleague Tom Taylor Kansas did this work. You can see in remarkably well-preserved cells a little branch bit of a fungus. Now if you compare that to the modern fungi in plant roots. So here's a plant root. There are hyphae, like I told you about, that are out in the environment the plant root, there are also hyphae in the cells of the plant. I'm going to show you an enlargement of this much better microgram. So you can see fungal hyphae at the top, like we've been talking about. And then you can see this remarkably branched hyphae. It looks like a small tree, so it's called a fungus, inside a plant cell. And what's really amazing is it doesn't break the membrane of the plant cell. The plant cell is alive. And there's a very large surface area between the plant cell and the fungus for the exchange of material. So the plant gets phosphorus and other minerals from the fungus, and it pays for that by supporting the fungus by giving nutrition. And so whereas a root could exploit a certain volume of soil, if it's got this fungus that's sending its hyphae out into the soil, it can exploit a much larger volume of soil and acquire enough mineral nutrient to grow. So they're old. If you compare those two, I think you'll agree, along with the rest of us, that that really is a fossil, a muscular fungus 400 million years old. The question is, are they important? Are they widespread? It turns out they are. 90% of plant species have this association. So if you look at a plant, you've got a 9 out of 10 chance that it's got a mycorrhiza in its root. What about the other 10%? Well, there's another kind of mycorrhiza, an ectomycorrhiza. They're not as common. Here you can see the hyphae, the white hyphae of this fungus surrounding a pine root. But they do the same thing. They provide nutrients to the tree, and the tree pays for it by giving the fungus sugar. And these are with oaks and pines. Now, our muscular mycorrhizal fungi, if you fertilize the plant sufficiently, it doesn't need the mycorrhiza. But with the oaks and pines and beeches, everything in the beech family and the pine family, if it doesn't have the mycorrhiza, it can't survive. It's an obligated survival system. So trees absolutely depend on these ectomycorrhizal fungi. So, I've told you about the 3 parts of the mushroom. That I really want you to remember. This is cocktail party mycology. When you're in a party and somebody asks you about fungi, I want you to remember, well, there are really 3 parts to it. And I've emphasized this thread, the hyphae, but now I'm going to tell you there are other shapes that fungi can take on. And so this is another shape. They can grow as yeasts. They're single-celled organisms. They're eukaryotes. They're just like the other fungi. In fact, many fungi that make hyphae can also make yeast. Some fungi can only make yeast. And I mentioned also at the start that, and what's interesting, they don't make the hyphae, they just make these single cells. And the cells can act as spores themselves. They're about, in a linear dimension, they're about 10 times bigger than a bacterium, so about 1,000 times the volume. Now, this fungus is actually far more famous than a mushroom. But I think most people, unless they've heard a lecture like this, don't initially understand that yeast is a fungus. But that's the other thing I'm sure. And in research, they've been very important. Two of these three fellows, Lee Hartwell and Paul Nurse, shared the Nobel Prize for Physiology or Medicine in 2001, working with yeast. Hartwell worked with Saccharomyces, the yeast of commerce, that we're going to talk more about, and Nurse with the different species, but both were yeast. And this allows me then to tell you about our colleague Randy Sheckler in the middle, our colleague in biology at Berkeley, who just shared the Nobel Prize for discoveries of the machinery regulating vesicle trafficking. And he worked with yeast too, with Saccharomyces. So in terms of research, it's a very famous fungus. But that's not why it's a famous fungus. The reason it's a famous fungus is it makes CO2, which is the basis of the leavened bread as it ferments. And it makes another product as it ferments. It makes ethanol. So it's the basis of alcohol and brew. So that is why it's really good. Now, I want to ask you some questions. There's a quiz in the middle. I want you to tell me what you think the total revenue is for these selected industries. Annual revenue. What do you think the annual growth is? Worldwide for alcoholic beverage. 200 billion. A trillion. 1,000 billion. What do you think it is for the automotive industry globally? Trucks, cars. now reveal there's one more shape in fungi. So this fungus here is a small fungus. It's representative of what we believe are what fungi look like in the earliest divergence. This is a pine pollen grain. So that's all the food that it needs. And this is the fungus. This is a young one. This is a nearly mature one. And these photos were taken before I made videos. So I don't have a video of it. Time-lapse photos. And this is seconds later, moments later, when the sporangium released its spores. And this is in the water. These are aquatic fungi. And what's interesting about them is they have their own power supply. They have a single posterior flagellum that lets them swim. They can also recognize gradients of nutrients and find new food supplies. Most fungi don't have this. Only these basal groups do. Now, if I give this, and given this talk 10 years ago, I would have said these fungi really don't interact with society. But in the meantime, one species, the Berkeley, has started killing amphibians. And it's just about extirpated the 2 species of Sierra yellow-legged frog. And we did some research on this some years ago, published in 2007 on the population genetics. And that showed 2 interesting things. One is that humans move this fungus because where the populations were mixed was along the Tioga Road. How the people come up to one lake and get a water bottle and go along and dump it at the next one. And also that it looks like it's undergoing sexual reproduction in nature. And if that's true, these fungi typically make resistant spores associated with sex. And it's been difficult to reintroduce frogs into lakes where the frogs have been extirpated. Probably because the fungus is still there. Now, it's bad enough that we're losing them in the Sierra Nevada, but it affects 30% of amphibians globally. It's a huge problem. And there's some people at Cal who are interested in this. One is a brand new assistant professor, Erica Bree Rosenblum, who's in our sister department, Environmental Science Policy and Management. And another is Dave Wade, who's an emeritus professor and was the head of our Museum of Vertebrate Zoology. He has been very important in organizing international efforts to combat the fungal disease, or at least track it. Now this flagellum, there's another interesting thing about the flagellum. That is that this structure, this flagellum, the single posterior flagellum, is shared between fungi and mammals in the form of mammal spermatozoa. This is a morphological similarity that it turned out was confirmed by molecular work. I'm showing you this. This is the publication that started molecular evolution studies for fungi. No other publications for other groups of organisms. And I put it up because my colleague Brian will tell you, this is the lowest form of scientific publication. It's a chapter in a book that one of the authors edited. So when you come up for promotion here, every 3 or 4 years you have to submit things. These aren't counted. This view is trash. But this one's been cited 12,000 times. And these are the guys who did it 20 years after the commitment. So, and this is my colleague Tom Brenz, who I'll talk about a little bit later. This is Tom. The guy who really did it was Tom White, who worked in industry, Cetus and Roche and Solera, and brought the polymerase chain reaction to our lab at a time that mattered. Steve Lee is a professor at San Jose State in criminology. And that work, that approach, allowed another group to make one of the most significant findings in terms of molecular evolution of fungi. And that is that fungi can link with animals. So that brings me to where fungi are. So here's a tree of life of organisms that have nuclei, the eukaryotes. The bacteria and the archaea aren't here. They're down where it says root. And there are fungi. And there are animals. And compare that to plants. So that was a shock to the scientific world. People had assumed that fungi were closely related to plants because they had cell walls, more closely than they were in the In fact, when I was hired at Berkeley, I was hired in the botany. Later it changed, now in the microbial division. So there was a last common ancestor between animals and fungi about 1 billion years ago. That's a long time, but it turns out we share a lot of biochemistry, certainly compared to bacteria. And so that fact makes it hard to treat fungal disease. In fact, the two targets that there are antifungal drugs for are a component of our membranes. Fungi have ergosterol, we have cholesterol. They're different enough that you can drug them, but they're not that different that you don't suffer the consequences when you take the drug. And also the cell wall. So they're hard to treat. And that's why there are all kinds of quack medicines to cure things like toenail fungi on the web, because there's no one good way to do it. And so I'm sure Zeta Clear kills the fungus, but it won't cure it, because you can't put it on there for the 6 months that you'd have to put it on for your nail to grow out to actually cure it. In fact, the good antifungal drugs, you don't want to take them for 6 months. You don't want to take them, your physician doesn't. So we still don't have a good treatment for things like that. So now I'm going to say a little bit about society. I've said a little bit already. I talked about these mycorrhizae, the ectomycorrhizae, bread and wine. One that I didn't talk about yet, that you probably have thought about, is that the first of the wonder drugs, the first bacterial antibiotic, came from a fungus, from Penicillium rudis. And it was these 3 people that created those drugs. Alexander Fleming, the famous one, he was good at research. But chain and fluoride were necessary for it to become a medical important thing. And I happen to have a personal connection with it. This fellow in the middle is David Atchison, a friend of mine in Berkeley. And this photo is just from about a week ago. And when he was 18 years old, in 1944, he was a U.S. infantry in the Second World War. And he got pneumonia in eastern France. And they gave him penicillin. And he came out of it. Now, he doesn't know if he would have come out of it if he didn't take the drug. But he's had a lifelong interest in penicillin since then. There are other ways that fungi interact with society. And they're not necessarily as nice. This is a friend of mine's house in Santa Monica. And that's a dry rot fungus fruiting in the door jamb. This house was bulldozed. It was so rotten. They also attack plants. So here's corn. Each little kernel is a fruit of the corn plant. And when this fungus, the corn smut, gets in there, it makes it into a tumor. And what do you suppose those black things that fill those tumors are? Spores. Lots and lots of spores. Now this is a particularly benign plant disease, because those tumors can be used to make an Aztec dish, huitlacoche. And so when you have huitlacoche, when you have a quesadilla of huitlacoche, it's got corn syrup. There's some less benign fungi that interact with corn. This is Aspergillus flavus. It's not really a corn disease per se, but it will affect corn, dried fruits, dried nuts, dried hard fruits, that either are insect damaged in nature or are improperly stored. What it does is it makes a secondary compound, aflatoxin, which is the most potent naturally occurring carcinogen. So quite a bit of your tax dollars go to the USDA to track and attempt to deal with this social problem. Now there are also fungi that cause human disease. So these are 2 lung cancers. The one on your left is healthy lungs. You can see the open cavities. And the one on the right is obscured here because of a fungus growing in it. It causes a disease called coccidioidomycosis, or more commonly, valley fever. Anybody here from Bakersfield? Anybody ever heard of it?
Yeah.
If you lived in Bakersfield, you'd thought, oh. So this fungus is a New World fungus, only found in the New World. It didn't evolve with humans because we've only been here for about 20,000 years. In fact, it evolved with these little beasts, and the reservoir for this disease is in small mammals, in the hot, dry areas of the New World. I want to go back. On this distribution map, see that? That's the Central Valley of California. So the life cycle, in my opinion, of this fungus starts with the spores at the top. They're hyphae, and make little internal spores. These spores are breathed in by the small mammal. And the mammal can mount an effective response, an immunological response with cells that hold the fungus in suspended animation in the granuloma, in which case the mammal has a long life. It's not effective. Or that little spore can grow about 1,000-fold, produce internal spores, release them, which then spread to every organ of the mouse, in which case it has a short life. But in either case, long life or short life, when it dies, the living fungus is in the animal. And it makes a lot of hyphae and a lot of spores, and the cycle persists. So it evolved with small mammals, but it turns out humans can get in the way. We breathe it in. We have the same possibility. Most of the time, we win. You have a long life, you may not even know you were ever sick. But in about 1% of cases, 10% of cases you go to the clinic, about 0.5% you're in good health. And this came to the news this year in June. This is ABC News. It's too small for you to read. It says, tiny spores are causing major headaches for the California Department of Corrections. So a judge ordered the Department of Corrections to move inmates from 2 prisons in California. There's also an issue of prison overcrowding. This is in addition to that, in Avenal and Coalinga. And the Coalinga prison is ironically named Pleasant Valley. That is a really good question. And the National Institutes of Health, I hope, are about to fund a study to try to understand that better. The only published work that I know of shows that African Americans have an increased risk for disseminated disease. And you'll see that here. So this is from the Bakersfield, California. Since 2006, 29 inmate deaths have been attributed to that. The disease is pegged as a secondary cause in 11 and a tertiary cause in 5 others. Of the inmates killed, 64% are black, 20% Latino, 16% white. So it affects, disproportionately affects Africans. And we don't know why. This guy, Royce Johnson, is the senior epoxy expert in Kern County. And he says the Fungi Valley prison has the highest rate of epoxy in the known world of fungi. He believes the prison should be closed. That's a big statement. So I'm interested in this fungus because it's changed from the normal thing that fungi do. Most fungi eat plants. Here's a brown nut fungus in the center. But this fungus eats animals. And so here is a group of fungi. This is a phylogenetic tree based on DNA variation. This is old, and this is the present. So time is moving slow. And on this branch, these are fungi that are associated with animals. The others are associated with plants. And so we were interested in knowing what makes them different, what makes them able to eat animals. And it turns out it's not that expensive to sequence the genomes of fungi. They're about 1% as large as our genome. So they're not that expensive. So we did some comparative genomics 10 years ago. And first, we looked at the genomes for genes that coded for enzymes that digest plants. So here, cellulose binding, tannins, pectin, pectate lights, pectinesterase. These are proteins, enzymes that chew up plant cell walls and feed. You can see that in this group, inference from this is that fungi, when the nuclei divide, they're losing genes all the time. Remember that hyphae had a lot of nuclei. And if a branch has a nucleus in it that lost a gene and it needs that gene, that branch just doesn't go anymore. So there's a selection to maintain the cohort of genes. But if the fungus has made a switch, in this case from plants to animals, and it loses a gene that's important to life on plants, it doesn't matter. There's a good chance that that nucleus will survive and make a branch and make spores. And that's what we think has happened here. That there's been no selection to maintain these genes. And if you look at the converse, we can look at enzymes that make genes, enzymes rather, that digest animals. And so here are subtolysins, peptidase, couple peptidases. These are enzymes that let you eat keratin. You can see there's an interesting thing. In this group of 4 fungi, one of them, histoplasma, has not had this gene family expansion. But the Coccidioides species and a close relative of theirs have. So again, when these nuclei divide, sometimes there's a duplication. That doesn't confer any advantage. That hypha that has that nucleus doesn't really go anywhere. But if it does confer an advantage, then it can be selected for it in case of positive selection. So we're interested in this, and we're interested in adaptation. And we'd like to know how the process occurs. But this occurred over at least tens of million years, maybe 100 million years. So, and it's obscured with a lot of noise. So we'd like to find a case where we can study it as it's just happened. In other words, we'd like to have one species or one population that's just recently diverged and then started to adapt to a different needs and we could do that with Coccidioides. We've done work on the population genetics of Coccidioides. We found that there were 2 species and at least 5 populations, and we could, for example, compare Central California and Southern California populations, or maybe populations within Central California, to look at this. And we thought about doing this. But Coccidioides is a tough fungus to work on. But we may get the opportunity. So in looking at adaptation, we're using as a model work that's been done with animals. And so I want to tell you a little bit about that. So this is the desert Southwest. This is southwestern topography, and these places are where lava has flowed over the desert. And this work was led by a man named Michael Nachman, who was at the University of Arizona until this semester. He's joined the Berkeley faculty to lead our department of zoology. And so here is the desert landscape of the lava flow. And so you can see that in a relative instant, a new environment's been created when it's very different. And it turns out the little mammals adapt to that. So here's a mammal with a coat of the sand. And here's a mammal with a coat of the lava, lives on lava. And if you put them on the wrong substrate, you can see that natural selection, driven by predation, would be very strong. They have the red coat. You're a hawk, would be no problem. So Nachman and colleagues then looked at the genes that make the element, the pigment, in the coat of those. And sure enough, there's evidence of natural selection, evolution of those genes in the population on the lava. So we thought that we'd like to do the same thing. And we would like to go with cocci, as I mentioned. We know about populations and may be able to find even further population separation. But as I said, it's a very tough fungus to work on. It kills you. You have to have a BL3 lab. My colleagues, people like my colleagues, wouldn't want that next to it. So that's where model comes in the talk. We picked a model fungus to try to learn how to do this. And this is a model fungus. This is an Neurostrum. These are cooked plants. They were in a fire. They weren't charred because they were killed. And you can see lots of little colonies of Neurostrum growing. Here's a blow-up. You can see hyphae, the hypha fungus. It's making lots of little spores. And we know that these spores function as male gametes. It's a sexually reproducible fungus. Right? It's a vegan, but it's not a raw food egg. It has to be cooked. This is the last of the 4 fungi that have won a Nobel Prize for something. These 2 guys, Beadle and Tatum, were professors at Stanford in the 40s and figured out that one gene made one enzyme, a big advance in genetics. So they got, 10 years later, they got the Nobel. So when we looked at our fungus, we were looking for an environmental factor like lava on sand. We really couldn't see it. And then we were looking for a phenotype like the dark coat, and we didn't see it. So we gave up. We tried various things. You have to do research where you have a chance of success and where you have a chance of getting money. So instead, we did something else called GWA. Now, acronyms are tricky, so if you Google GWA, but the one we want is Genome-wide association. So this is an example of genome-wide association in humans. So what you do, you're trying to associate phenotypic variation and genotypic variation, genetic variation and phenotypic variation. Here the phenotype is diabetes. So you get 1,000 people with type 2 diabetes and 1,000 without. That's the phenotypic variation. And then you sequence their genomes and you look for genes in which the diabetic group has a monoallele and the non-diabetic group has a different one. So then there are statistics involved because it's not quite that simple. It's never all or none. But it would show you that there's an association between specific genes and the phenotype of this disease. So we thought we would do this with our fungus. And when I say we, I mean Louise Glass, who's nice movie I showed you earlier, a colleague of ours in our department. Rachel Brown, who's an expert in this kind of analysis in the Department of Molecular and Cell Biology. And then the people whose hands actually did the work, Charles, David, and Julie, and Louise's lab grew the fungus and made the libraries and sent them off to active sequence. And Chris Ellison, who was a grad student with me, did the analysis with a lot of help from Rachel. And we picked a population of neurosperm, one clade, one species that surrounded the Gulf of Mexico, because it had a really well-sequenced individual that we could compare to all the other individuals that we were going to sequence in this fungus. It cost us about $300 to sequence a fungus now, cheaply if we have a nice sequence to compare it to. So you could do 100 for $30,000, which is about half what a graduate student costs per year, or half what a postdoc costs. So it's really relatively, in the funding of science, it's very inexpensive to do. We picked it because it had this nicely sequenced individual. And you'll see that's important as we go later on. So here are the fungi we're interested in. When we had 50 genomes, we're aiming for 100. We stopped and analyzed. So now here is where luck plays a part. The first luck is bad. So if you want to do GWAS, genome-wide association in humans, you want to be sure you're just dealing with humans. You have to do it in an interbreeding population. If by mistake you included some chimps and you looked at the population, you would find phenotypic differences between humans and chimps. There are a lot of them. Ability to swing in trees. And you go, okay, that's my phenotype. It's going to be the first one I'm going to look at. And you'd find that it is associated with every genetic difference between humans and chimps. And there are 35 million single nucleotide polymorphisms, nucleotide mutations between chimps and humans. It would associate with every one of them. You'd be no further than when you started. Every gene would associate with all of them. So it's very important that you know what your population is. So when Ellison analyzed the data, he found that we didn't have one population. We had 7 populations. So that's the bad one. That put genome-wide association on hold. We eventually got there. We have 112 population. But the good one is that 2 of them, a Caribbean population here, and a Louisiana population, had enough individuals that we could do some analysis. And the analysis that we could do is the one we wanted to do. So now we had 2 populations we could study adaptation. So we weren't funded to do that. We never could have gotten that. But by bad luck and good luck, we had the data. And it turned out that those two populations were separated. The Caribbean population was south of the Louisiana population, so the latitude. And it's colder in Louisiana in the winter than it is in the Caribbean. It gets just as hot in summer, but it's colder in the winter. So what Chris did then was to line up those 50 genomes and scan through them, 500 base pairs at a time, and look for regions that were exceptionally divergent, exceptionally different. And our thinking was if they were exceptionally different, it could be evidence that some region of DNA had come into the species, into the population, from another population by hybridization at the margin, then repeated mating of those hybrids back in the population. And if there was a gene in that region that conferred an advantage that let them have more progeny, more babies, it could spread through the population. So there are 7 chromosomes in neuroscope, so he scanned through these chromosomes. And there are 3 ways that he scanned. And it turns out each of them is susceptible to artifacts, but to different reasons. So we reasoned that if we found a region that was found by all 3 methods, it might be good. So there are 2 such, one on chromosome 3 and on chromosome 7. So here's what they look like, and this takes a little explanation. There are 4 colors, because there are 4 nucleotides in DNA, A, C, G. The columns are single base pairs, single nucleotide positions. They're all multicolored because I'm only showing you the ones that are variable. There are 400 times as many that are invariant because these are really closely related. But if I put them on there, it'd be 400 times bigger than you could cover. So I threw out everything that's not good. These are only the variables. That's the columns. The rows are the individuals. So they're the Louisiana individuals, the Caribbean individuals, and individuals in those 5 of the populations. And what I want you to see is there's a sharp line in this island of divergence between Louisiana and the Caribbean. And also that within Louisiana or within the Caribbean, in many cases, there's not much variation. That's what we would expect if a piece of DNA came in from another population and spread rapidly through Louisiana or through the Caribbean. It shouldn't have much variation because there hasn't been time for mutations to occur. And they should be different. So compare that to the rest of the genome. You don't see that sharp. And where there's variation on one side, there's variation often on the other side. So these are part of that ancestral population. What we think is the Louisiana group is the pioneer. It's moving into colder regions. This is a tropical and subtropical fungus, usually not found in colder regions. And that this adaptation may have allowed it to do so. So if we're right, there should be genes in this region that confer an adaptive advantage to growing at low temperatures. So here are the 3 genes: phospholipase, dead box RNA helicase. And this is just a gene number because it makes a protein. The protein's in the mitochondrial outer membrane, but we don't know what it does. But the one that caught our attention is this one because it's induced by cold shock. The RNA helicase protects against cold shock. This is the other region, the chromosome 7 region. You can see the region of divergence. You can see it's a little more complicated, and there are more genes there. And the one that caught our eye is this, a prefold. Most chaperones deal with high temperature problems that proteins face, but this one deals with cold sensitive proteins, the actin cytoskeleton and cytomusculature. So again, on this group, the temperature was involved. And you remember there's a temperature difference. So what we did then to test this hypothesis, it's easy to make hypotheses, but unless you can test it, they're not worth much. We took 10 individuals from Louisiana, 10 from the Caribbean, and we grew them at normal temperature, 25 degrees Celsius, and the low temperature, 10 degrees. So here's the growth at 10 as a fraction of the growth at 25 to control for individual differences in this. And here's Louisiana and the Caribbean. And you can see here that Louisiana grows on average better than the cricket, and it's statistically significant difference. You can also see that they don't like it at temperature. They grow 1/5 as fast. The Rossberg grows very happy at 30, lower higher temperatures. Remember I said it's tropical and stuff. So having survived that, we thought we should look at individual genes. Now, the other reason for picking this group of Rossberg, in addition to it having a nice genome sequence, is that other scientists, another group of scientists, remember it's a model organism, there are many people who study it, had gone through and knocked out every gene that can be in this fungus. So we could send an email and get a collection of strains that had each genes. So we did that. So here are the genes that have been knocked out. Here's the wild type, no knockout. And this is growth at 10 degrees Celsius. So we did a quick and dirty experiment. There are two things I want you to see. The first is just knocking out a gene doesn't necessarily affect its ability to grow over temperature. If every gene that we knocked out affected that, then it wouldn't be a good experiment. It would just be somehow the trauma of having a gene knocked out affected. But some of them look to us, 3 of them, that outer membrane protein, the helicase, and the prefold, looked as if they might actually be involved in adaptation to cold. So we did a better experiment. We mated the knockout strains with the wild type, and we picked progeny that had either the knockout allele or the wild type. And we grew them did the better experiment, grew them at 10 as a fraction of 5. The helicase showed a difference. The wild type allele grows better at low temperature than the knockout. That's significant. And the same for the prefolded. It's a more dramatic difference. It's also significant. The wild type grows better than the knockouts. So our hypothesis has survived these tests. Now, as Brian will tell you, you can never prove a hypothesis. You can only disprove it. So we haven't been able to disprove it so far. So we think that these two genes, at least, are involved in adaptation to growth at lower temperature. So when I started talking about this a few minutes ago, I said we didn't know what the environmental factor was. We didn't know the phenotype. We didn't know, certainly didn't know the genes. We went about it completely backwards compared to what Michael Nachman did with his mice. We looked for the genes first. We found the genes first. Then we guessed at the environmental factor. And then we could test that with growth as a phenotype, a surrogate for fitness. So we call this reverse ecology. We think it could be very useful for microbes in general, where it's very hard to guess what's the environmental parameter that matters to this microbe, and then what's the response to it. I want to point out, too, that it could actually also be useful in the mammal studies. And Michael would agree with me. I've had this discussion. When they saw this difference, they looked at genes, they're called candidate genes, genes that they knew were involved in melanin synthesis. There may be many other genes in the mouse that are involved in adaptation to life on the lava. I'll bet it's a lot hotter on the lava than it is on the sands. So if he did our approach, which I'm told he's now going to do, sequence a bunch of these mice, look for the differences first and then work backwards to find if the genes are important and work backwards. He might find that there are other genes that are involved in adaptation. So we're hoping to get to try this with coccidioides. I hear from Dennis Dixon at the NIH that we will. It's possible. We're doing some other things with this approach. With the Genome-wide association approach. So we're involved with the Energy Bioscience Institute, and they're interested in turning sugar into biofuels. They're interested in turning plant cell walls into biofuels. This is a pile of sugarcane cell walls, sugarcane bagasse. And this is a couple of people here, a former postdoc and a worker at the sugar mill, getting very hot compost out of this large pile of bagasse. And in that, we found some yeast that are thermophilic. So we're hoping to do some work on that. Here's one of those yeasts. And they can ferment at high temperature, which would save money and make them mild. And the people involved in this, my colleague Tom Brunson and I are involved in this. And this is Chris Somerville, who's the director of the Energy Bioscience Institute at Berkeley. And as you leave today, if you look across Oxford, right, at the southwest corner of Oxford and Paris, you'll see a big new building that Chris is located with. Several members of our department are in this operation. We're also interested in applying this to ectomycorrhizal fungi. I mentioned those earlier. And the postdoc who's working on this is Sarah Bronco. And she has isolated strains of this little ectomycorrhizal mushroom, Scrollus brevus, which is very common in California, but unfortunately not worth eating. So she's got them from the coast, and she's got them from Sierra Nevada. And she has sequenced the genomes, and she can see that there are two populations, a Sierra one and a coastal one. And so now she's scanning through those, looking for regions of interest. So, to sum up, I've told you a little bit about fungi. You'll remember that there are 3 parts to a mushroom. Fungi can be filamentous and have hyphae. They can make yeast, and they can also have their own motility. They can make zoospores. I've talked about where fungi are in the tree of life. Their closest big kingdom relatives are animals, and they're more distant from plants. I've said a little bit about society. I should have emphasized, but I have an opportunity now. Fungi are in their living, they eat dead things. They're saprobes, wood decay, like the guy's house, or in fact yeast, working on sugar to make ethanol. That's a decay fungus. They can be symbionts. They can work with another form of life, and that can range from being a positive symbiosis, like the mycorrhizae, or in our view, a negative, like a disease of humans or a disease of plants. So those are the 3 ways that they live: saprobes, parasites, or symbionts, mutuals. And then I said a little bit about model fungi. I told you about yeast, certainly the most important model fungus, Neurospora, the most important filamentous model fungus. And then I also mentioned penicillium, which is really not a model fungus, but it has produced the important animal. Now there's money involved in research. Thank you for your tax support. The National Institutes of Health funded that work on coccidioides and actually coordinated the work on Neurospora. It also funds a stock center that keeps these fungal cultures so that I can work on the fungus that some previous scientists have worked on and continue his or her work. Without that, if those stocks are lost, then old research that we all paid for goes away. The National Science Foundation, which funded a lot of the work on Neurospora, and also helps support the phylogenetic stock center. And then at Berkeley, a former chancellor, Cheng Lin-Tian, has a fellowship named in his honor, and that supported Chris Ellison's research and also Tom Shotkin's research on coccidioides and on Neurospora. And finally, this is the Berkeley Mycology School, which is about 10 years old, even though Berkeley Mycology is more than 100 years old. And with that, I'm happy to take any questions. The first question